Hello everyone, my name is Hsieh, and I'm a PhD student in Center of Molecular Medicine in Norway. Today I'm going to share one of my research topics, adjustment of spurious correlations in co-expression measurements from RNA-seq data. Before we do expression analysis on RNA-seq data, it's relevant to perform data normalization to remove the technical variability. Most of the normalization methods for RNA-seq data uses global properties to infer the scaling factor or normalization factor to correct for the technical variability. However, when the source of the technical variability is unknown, it might be difficult for the normalization method to tell whether the variability is caused by meaningful biology. Recently, a quantile-based normalization method is proposed. The smooth quantile normalization is specifically designed to address this issue. Smooth quantile normalization allows the users to provide the information of biological groups into the normalization methods, and the algorithm will try to preserve the variability across different biological groups and remove the technical variability. The method also shows better control over the within sample variability. However, we have found that such methods might introduce spurious associations between lowly expressed genes. We've dived into the algorithm and found that this is because the algorithm takes the average of the normalization factors for the corresponding quantile if the recount is the same for different genes. As normalization factors are always a positive number, the average will deviate from the mode for lowly expressed genes, and this small added variability would introduce false positive associations. And you can see it from this small example for the orange and the purple entries on the bottom right side. And one obvious way of addressing this issue is to just take the median of the normalization factors instead of the mean for the corresponding quantile. And when there's a large differences in the sequencing depth, this is more likely to happen. For instance, data set collected from different sources or tissues, such as the GTEx dataset or the ENCODE dataset. And for the data set that has already been quantile normalized, we developed a tool called Kaiman to correct for the normalized expression. The Kaiman algorithms first extract the normalized expression for each group, and then sort the expression for each sample. And from there, we use the sorted result to infer what would be the average of the normalization factors. Noted that the quantile before and after the normalization will stay the same. And then we use the average of normalization factors to infer the distribution of for the normalization factor corresponding to different quantile using a mixture of Poisson distribution. Lastly, we use the median of the Inferred distribution for the corresponding quantile to adjust for the normalized expression. Here are some of our results. On the left panel, it describes how we infer the distribution of normalization factor with mixture of Poisson distribution. On the right panel, we show that Kaiman would reliably recover the true distribution of the normalization factors. We then define tissue-exclusive genes the genes that exclusively express in one of the tissue to validate whether or not our model can effectively remove those false positive associations. We can see that the false positive associations between genes expressed in different tissues are removed after Kaiman correction for the GTEx dataset. For the ENCODE dataset, we use the spike-in genes to construct a validation dataset and compare the gene associations with our validation dataset. We can see that Kaiman can improve Q-smooth results for both tissue-exclusive genes and random genes. Lastly, we show that Kaiman can improve the downstream sample-specific co-expression network analysis. This figure shows the difference, more specifically the log-fold change between the edges of co-expression network compared with the ground truth co-expression network. We can see that after Kaiman correction, the network we constructed is much more similar to the one constructed using the ground truth dataset. So to wrap our research up, we found that first, quantile-based normalization methods might introduce technical variability that leads to false positive association between lowly expressed genes. And this is because of the average of the normalization factors corresponding to genes with the same read count. And we've developed Kaiman that use a mixture of Poisson distribution to effectively correct for the normalized expression and remove the false positive associations. And thank you so much for listening. I'm looking forward to discuss with you in the poster session.